Thank you, Faz, for your kind introduction, and I'm so glad to be here. So, kind of the previous talk, we kind of listening about the disease biology, and this time we're going to switch to the population-based science. It's pretty straightforward. So, majority of the factors we kind of encounter every day. So, today I just want to briefly introduce some work we have been done, and we're kind of continue working on this, and most of them has not been really finished yet. So I just share some work we have done so far. So weird. So as you all know, the lymphoma, there's kind of two basic big groups of lymphoma. One is Hodgkin disease and another is non-Hodgkin lymphoma. And for the Hodgkin disease, the mainly affect the B lymphocytes, and the main diagnosis age is around 33 years old. It's pretty younger people. And for the non-Hodgkin lymphoma, it's a whole kind of big group of diseases, and it can affect both B lymphocytes and T lymphocytes, as well as natural killer cells. And for the non-Hodgkin lymphoma, the mean diagnosis age is pretty old. It's around like 60 to 65 years old. So as you know, there's quite a lot of different histological subtypes of the non-Hodgkin lymphoma, and they kind of arising from the different stages of the lymphocytes maturation. So you know, the non-Hodgkin lymphoma subtype classification system has been changed over time. There's a very complex, different, classification system. Actually, that makes for the epidemiologist, or both for the clinician and the epidemiologist, very hard. It's very challenging to compare all those results. Because previous study using some different kind of the classification system, it's hard to compare all those different subtype results. And since 2001, there's WHO kind of introduced a new WHO classification system, so which is right now kind of recommended for both clinical setting and for the epidemiology study. So as we know, the most common non-Hodgkin lymphoma subtype is diffuse large B-cell lymphoma, which account about like 30%. And the second most common one is a follicular lymphoma. And for all others, it's pretty rare, and some even only can less than 1%. So the next few slides, I just want to show you the the overall picture about the incidence risk pattern around the world and also in the United States. This slides show you the global incidence of non-Hodgkin lymphoma. You can see there's the disease rates vary from the different places around the world. You can see the highest incidence rates appear on the more developed country. And for the developing country, the incidence rates is pretty low, like China, Asian, and Africa. So this, as you know, there's a WHO, there's one institution that belongs to the WHO, the International Agency for Research on Cancer. They periodically publish this incidence in 5 continents, and they're collecting all the tumor registry data around the world, different country, and they publish the data, kind of give us an overall picture, what's the incidence rates for the different cancer type around the world. So from those incidence pattern and the trends, you can, make sense which one is kind of increasing, which one is decreasing, and which one is kind of remain stable. And from that, you can generate a certain hypothesis, or right now we should focus on what. And this is also no matter, around the low incidence country or the high incidence country, you can see the non-Hodgkin lymphoma incidence has been increasing since the 1970s. So this just, I selected a few countries from the different continent and just represent different continent. You can see every single country almost has an increase in incidence for non-Hodgkin lymphoma. So this is the data from the United States, the SEER program. So the tumor registry program is very popular right now in the United States. And I think almost every cancer researcher is kind of familiar with this program. And you can see from the 2001 to 2005, this is the most updated data sets. And you can see the non-Hodgkin lymphoma among female is the 5th leading cause of the cancer. And among men, this is the 6th leading cause of cancer. So even though there's a man, the absolute rates for age-adjusted incidence rates is higher among men compared to women. It's about 1.5 times higher. So this is the time trends in the United States from the SEER data. So as you know, the majority of the SEER program is starting from the 1973, except the Connecticut Tumor Registry is one of the oldest ones starting from 1935. So only from the Connecticut we can see the really long-term time trends, but for the entire states you can only see starting from the 1970s. And you can see the incidence rates for the non-Hodgkin lymphoma, both men and women, they showed increasing pattern until the late 1990s, kind of starting pretty stable. And for the mortality of non-Hodgkin lymphoma, they showed a constant increase until the 2000. There's the new development of the new medication to treat the non-Hodgkin lymphoma, so they significantly reduce the mortality rates. And as we talked, there's a whole group of the different subtypes of non-Hodgkin lymphoma. And from the SEER data, you can see from the time trends, you can see the different non-Hodgkin lymphoma subtypes, they showed very different incidence trends pattern. And also from the, the earlier epidemiology study and also most current epidemiology study, we also found that there's for the etiology for different histological subtypes is also different. And that's why, at the beginning for the epidemiologists, so we all kind of treat the non-Hodgkin lymphoma as a one single disease, and we look at the pattern, and we look at what's the risk factor causing this non-Hodgkin lymphoma increasing trends. And afterward, we all realize, we cannot treat this disease as a one single disease. We have to look at a different subtype, different type. So every single subtype, they might have one single disease, they compose a different pattern, and they might also have the different etiology. So this is in the 1991, there's a National Cancer Center kind of sponsored a meeting, and they also issued a special issue on the cancer research. They called it the Emerging Epidemic of Non-Hodgkin Lymphoma. So you can see from this picture, they showed the starting from the SEER data from the 1970s until the later 1980s. During the past 2 decades, there's constant increasing of the non-Hodgkin lymphoma incidence. And before, the non-Hodgkin lymphoma is a pretty rare disease, and until then, the non-Hodgkin lymphoma is pretty common among the general population. So they want to figure out what's the reason cause this rapid increase of non-Hodgkin lymphoma. And actually, the one thing people realize, because the non-Hodgkin lymphoma is kind of affect your immune system, and the epidemic of HIV infection and AIDS might account for some certain increasing trends. But it turns out, the AIDS infection and HIV infection AIDS patients, the prevalence in the general population about this group of people, it's not that high. And for those large increasing trends, you cannot totally attribute it to this increasing prevalence of the infection of the HIV and AIDS. So they consider there must some other environmental factors might also contribute to this increasing trends. So during that meeting, they tried to, kind of determine whether those increasing trends is real or because due to the detection bias, some overdiagnosis. And after the meeting, they decided this increase is really real, and they need to figure out, what factors and which might be responsible for this increase in trends. And actually, since then, there's quite a lot, several big population-based case control study has been launched in the United States. And one of the studies kind of from the Connecticut, the PI is Dr. Tongzhen. And, so far, what do we know about the non-Hodgkin lymphoma, the risk factors? So there's only very few established risk factors. You can see all those established risk factors is kind of related to the immune dysregulation, like previous talk about the HIV/AIDS infection, and some, the organ transplantation patients, they kind of obtain those immune suppressant medication treatment. And also for the certain virus infection that's only related to the certain subtypes. And also some autoimmune diseases which relate to the non-Hodgkin lymphoma. And all those kind of established risk factors which can only account for a very small portion of the diseases. So that means lives a vast majority of the NHL cases unexplained. So we don't know what exact risk factors can cause those non-Hodgkin lymphoma disease. So the following, I just want to first introduce this is the population study we kind of conducted in the Connecticut, and the PI was Dr. Townsend. And this study was conducted in the 1996 to 2000. And initially, because during that, I mentioned that meeting, during that NCI meeting, and one of the major suspected risk factors is hair dye use. So the reason is there's a kind of, from the Iowa and Nebraska study, that there's a kind of big, huge population-based, multi-site population-based case control study conducted by the NCI people. And from that study, they found that it's really strong signal for the personal hair dye use, kind of related to the non-Hodgkin lymphoma. And then this, the personal hair dye use, people say that that's only applied to your hair, why the kind of cause non-Hodgkin lymphoma. And those hair dye can really kind of absorbed by your scalp, and they can get into your body. And actually, during the 1980s, early 1980s, there's a kind of NCI did several animal studies. They found certain ingredients in that hair dye formula is were carcinogen, and they kind of FDA kind of proposed a regulation. They announced all those, the hair dye company. So if you want to sell your hair dye, you have to put a label on your hair dye products. So this is a kind of potential carcinogen in the hair dye. So the hair dye company won't do this, because if they put the label, nobody going to use their hair dye, will really affect their sale. So that's why they kind of make the decision. They changed their hair dye kind of products. They removed all those carcinogens that has been identified as a carcinogen by NCI, animal studies. And then right now, the current market is kind of all new products. And during that time, we proposed this study, try to, see whether this hair dye use is kind of really related to the non-Hodgkin lymphoma. So we all know the hair dye is more popular in women. So the prevalence in men using hair dye is pretty low. So that's why our population is mainly focused on women. We did not include men in this population. So we can identify the cases from the rapid case estimate, which is affiliated with the Yale Cancer Center. And we identified more than 800 cases which are eligible for the study, and it's only 600 agreed to participate in the study. And we did an in-person interview collecting all those exposure data, and also collecting their blood samples and buccal cells to do the genotyping and for the gene analysis. And we also identified 700 more than 700 population-based controls. We use two different methods to recruit all those population controls. For the people aged under 65 years old, we use a random digidialing. Kind of just randomly dial their phone number and recruit them as our potential controls matched within the age group, 5-year age group. And for the older people, we use, before they called HECFA database, and we randomly select those older people to as a controls. But right now, they're not allowed, this HECFA right now, they call this Center for Medicaid and Medicare Service. They're not allowed us to using their file to do the study. So right now we have to no matter it's older people or younger people, have to rely on the random digidialing methods. So anyway, for this study, we recruited 600 women and about 700 population-based controls, and both of them did an in-person interview and collecting their blood sample, everything. So before we move on to the results, I just want to introduce a little bit about the InterLymph. This is the International Lymphoma Epidemiology Consortium. So this InterLymph has been established in 2001, and the reason for right now a lot of people knows there's a lot of consortium everywhere, almost for every single diseases. And when we initiated this InterLymph consortium, because after the 1990s, there's quite a few epidemiology study has been launched in the United States. And more and more we realize, we have to look at the different histological subtype. We cannot just treat them as one single disease group. So actually that brings a lot of challenge, because when you break down with the different histological subtype, so every single individual study, you will have very tiny little numbers. So you will not have enough statistical power to really look at all those associations. So that's why we the researchers from all over the world together we try to form this International Lymphoma Consortium, and starting from the 2001. Right now it's pretty successful. We have many centers joined this lymphoma consortium, and the major goal is, for all the individual study, when you find some signal, publish some interesting finding, you can replicate, within this InterLymph, and including all those, lymphoma study, and you will get enough cases and controls. You can look at your association more precisely. So from this, so we talked about the personal hair dye use is kind of from our study. I just want to first show you what's the hair dye results we found. Actually, from our individual study, we do find the increased risk for personal hair dye use is related to the non-Hodgkin lymphoma, and particularly for the follicular lymphoma, and particularly focus on the women who started using hair dye before 1980. From our own study, we did not find a women who used the hair dye after 1980. But and then we kind of using this InterLymph database, we did a pooled analysis, which included more than 4,000 cases, more than 4,000 controls. And then from this study, we really replicate our results, and we got the very similar results. And there's an increased risk mainly found for the follicular lymphoma, and another one is chronic lymphocytic leukemia and small lymphocytic lymphoma. And both mainly found it for the people women who started using hair dye before 1980. But from this pooled analysis, we also found a signal for the women who started using hair dye after 1980. So right now we really cannot really see that the current products is really safe products. But there's a kind of very interesting story. So the international, the IARC, International Research for Cancer, International Agency for Research on Cancer, they have the monograph. They kind of periodically publish this monograph and specifically focus on all the different kind of chemicals or certain lifestyle factors. They would define whether this is a human carcinogen or not. And they organized a meeting for the hair dye and other, industry-related dyes. And I think it's last year, the beginning of the last year, they organized this meeting to try to define whether the personal hair dye use is a kind of human carcinogen. And during that meeting, one interesting thing is, because even though we have various some publication and provide some evidence for those hair dye use related to the non-Hodgkin lymphoma in the epidemiology study, but there's no data available from the benchwork side. There's no toxicology really did the toxicology study related to they buy hair dye, do some experimental study. But first of all, it's really hard, and second of all, it's pretty sensitive. During that meeting, actually, there's 2 people from the big hair dye company sitting there observing what we're talking, what we're discussing. And they constantly provide us, all those publications they can find, and many publications actually they did from their company. And it's really hard. The hair dye ingredients is millions ingredients in there. So you cannot just say one ingredient it's not a carcinogen. You will say this is safe. That's not the case. So the hair dye, you also when you apply the hair dye, you will have the oxidization process when you combine all the different ingredients together. This kind of probably will produce some more kind of different, and who knows what's new chemicals will be produced. So it's very hard to do the experimental study. So that's why during that meeting it's a kind of debate is really hard with the people. And we don't agree to each other. So even though within the epidemiologist and within the toxicologist and with the biologist, nobody kind of is really agree to each other. So and it turns out we still consider this as a kind of potential possible, but it's not the human carcinogen. But anyway, from the from our study, we do find some this kind of increase in signal. Because this hair dye use is very popular, the prevalence is very high among the general population. So even though the very small increased risk for the population attributable risk is could be reached very high. So I just simply calculate the population attributable risk percentage. So we could see this could be explained as if there's a personal hair dye use is a cause of the follicular lymphoma. So in the general population, the 18% follicular lymphoma or CLL/SL will be kind of attributable to personal hair dye use. So that's kind of the interpretation for this population attributable risk percentage. So the next environmental factors, cigarette smoking. So cigarette smoking in the non-Hodgkin lymphoma, we really did not find a very strong kind of relationship. It's very similar with the hair dye, but even kind of weaker compared to hair dye use. And they only found for the follicular lymphoma and for the current smokers. And the population attributable risk is around like 8%. So we also looked at occupational exposure for the solvents. And we look at all solvents combined. We kind of using the occupational exposure assessment matrix. So we, from the questionnaire, you ask them which chemical you're exposed to. I don't think the general population will be able to answer that question because they have no idea what that, especially, and we just ask them, what's your occupation title and what industry you have been working for. And we kind of collaborating with the National Cancer Institute, the occupational study group, and they're using their job exposure matrix to link those, based on their job title and work experience and how many years you work there. So we kind of figure out what chemicals they have been exposed to. And we look at the solvents exposure, and you can see there's a pretty strong association found with the formaldehyde and some organochlorated solvents. So you can see that mainly we found for the diffuse large B-cell lymphoma this time. And from this, we also kind of calculate their kind of population, general population attributable risk. You can see compared to the previous 2 environmental exposures, this exposure have much stronger association with the non-Hodgkin lymphoma, but their prevalence in the general population is pretty low. It's not compared to the hair dye smoking in the general population. So a lot of people kind of using hair dye and smoke, but for the occupation exposure to those chemicals is relatively low. So even though they have the pretty strong association, their population attributable risk is relatively low compared to the previous 2. So because of the time limit, I'm not going to talk everything. So just to give you some kind of taste about what's the environmental factors, what we look at. And we also looked at many others like other pesticides exposure, physical inactivity, and HCV, hepatitis C virus exposed, and sun exposure, and also the nitrate exposure, and also the dietary. It's a very interesting one. And we do find a lot of very interesting findings. I hope in the future we have some opportunity to share all those results. So as we all know, when you look at the pure environmental factors, people will see everyone use that, like lung cancer, smoking. A lot of people are smoking, but it's not everyone get lung cancer. So that's the same thing. There's a gene-environmental interaction exist. So every single environmental factors, you have to kind of interact with your body's, genetic susceptibility. If you're susceptible to these environmental factors, you're better to avoid those environmental exposure. So that's kind of our future direction, and also currently we're working on this, do all the gene environmental interaction to look at all the different environmental factors and with the different genetic polymorphism or even the phenotype measures, so to look at which, genes play a, stronger role with those interact with the environmental factors. So from those gene-environmental interaction results, we can really truly identify which one is a high-risk population. So we can really put our intervention strategy to those high-risk population. So this is all the investigators has been involved in this study, and we also actually did a lot of genotyping work, and we looked at all the many different genetic pathway polymorphisms related to the disease. And currently, we also get several gene-environment interaction paper out. And also, again, we also want to utilize all those information to look at whether those environment or genetic factors related to the non-Hodgkin lymphoma survivorship, whether they're also kind of related to the non-Hodgkin prognosis. So I just want to thank all those investigators who have been put a lot of effort on this project, especially from the PI, Dr. Tongzhen, who initiated the study, and also the many others. Ted Hoffer is our best statistician, and he also gave a lot of advice in the data analysis, and also many others, and also from the National Cancer Institute. We have been collaborating on the project. Okay, thank you very much.